Yeah. Hi, everyone. My name is Sindhura. I'm a graduate student from Virginia Tech, and my paper focuses on prediction of gene regulatory connections with joint single cell foundation models and graph based learning. To give some background, I want to talk about gene regulatory network inference from single cell transcriptomics data. We've heard this in the morning session as well, but just for the recap, uh, gene regulatory networks describe how genes interact with each other, and they are a very complex system of interactions between transcription factors and the target genes. Each of these networks can be represented as graphs, where nodes in the graph represent genes and edges represent regulatory relationships. As the transcriptomics techniques are advancing, the growing availability of seRNA-seq data um, enables the study of cellular diversity by enabling gene expression profiling at single cell resolution. And one of the most critical applications of seRNA-seq data is the GRN inference, which plays a crucial role in understanding cellular processes such as differentiation and development. Since past 20 years, many computational methods have been developed to infer GRNs. However, accuracy and reliability is still a significant challenge due to the high dropout rates, sparsity, and noise that is inherent in the single cell expression data. So, earlier methods employed traditional unsupervised methods. So, earlier methods like Genie3, GRN Boost use techniques like tree based regression to identify sets of genes that are co expressed together. But given the large amount of genes that are profiled, some of these co expression signals or statistical associations may arise purely from chance. Also, the rapid growth of experimentally validated TFDNA binding data has enabled the development of supervised learning approaches where the models learn from known regulatory interactions to infer novel regulatory relationships by framing it as a supervised link prediction task. More recently, graph based learning frameworks such as GeneLink and GNNLink have demonstrated promise in modeling the complex interactions within GRNs. And graph neural networks, which are used in these models to encode topological features of regulatory networks, are inherently designed to handle handle the complexities of the graph data, but higher performance of supervised learning methods, especially when we are training large parameters or deep learning models, large amount of experimentally validated GRNs is required as a labeled training data, which is often experimentally expensive to collect and limited in amount. So, graph learning alone might not solve the problem, even if we frame it as a supervised regulatory link prediction, which is where large scale pre trained foundation models could offer a transformative opportunity to address this gap or challenge. More recent studies inspired by the large scale pre training of large language models, which are foundation models that learn from the general patterns in language and can then be fine tuned for other fine tuned for other task specific data sets. So models like or papers like SC-BIRD, Gene Farmer, SC Foundation that have been pre-trained on large-scale single-cell transcriptomics data have shown promise in interpreting the languages of cells, thereby providing rich informative gene representations for advancing network biology. So, what is a single cell foundation model? We define it as a model that has been pre trained on unlabeled sRNA-seq data spanning millions of samples, which is how it is able to learn these informative representations. So these models are based on transformer architecture that harness the self-attention mechanism to maintain attention over the input space of each transcriptome. This attention and transformer architecture allows the model to learn contextual representations for a particular gene in a particular cell based on the expression profile of the surrounding genes or the neighborhood genes in the same cell, giving us a very contextualized latent representations for genes. And these are usually trained using a self-supervised pre-training objective. In case of large language models, it's usually masked language modeling, but in this case, it could be masked gene expression prediction or masked gene rank value prediction based on the type of foundation model that we choose. So, essentially, the research question we are trying to address is, can we leverage this contextual information from single cell foundation models or SEFMs and the graph learning and structural patterns from graph learning to improve gene regulatory link prediction? For this, we propose a framework called SERegnet. Here is a schematic overview of our framework. And SERegnet integrates gene representations from foundation models and GNNs, and these combined representations are then fed into a classifier for gene regulatory link prediction. As shown in the figure, the foundation model parameters on the top in blue are frozen since they are more expensive to train, and the graph encoder or graph learning in the bottom section is trained end to end using the joint graph learning for supervised prediction task. More formally, the input RNA-seq data is represented as a cell by gene matrix. So in this study, we focus on cell type specific GRN inference. So we focus on using cell type specific benchmark data sets to infer cell type specific GRNs. So for each benchmark set, the cell by gene matrix is represented as an N cross T matrix, where N is number of cells and T is number of genes, and each element represents the RNA abundance for gene T in cell N. Now, uh, this input is used to construct the initial representations for the single cell foundation model that are then processed through L successive transformer encoder layers that will give us final contextual gene level embeddings as output. Now, this L could be different based on the choice of foundation model that we use, where L is the number of transformer layers. For each cell type specific benchmark dataset, once we have the gene embeddings for all the cells, we use mean pooling across all the cells in a specific benchmark dataset to get a balanced gene level representation that encapsulates the average gene activity throughout the entire dataset. Digging deeper, the foundation models we use in our study are we pick diverse set of foundation models, namely SE-BERT, GeneFormer, SE Foundation, which are inherently different in the way they model their input, their model architectures and training procedures. Specifically, the input design and pre-processing steps vary for each model as detailed. So, for example, in SC-BERT, the number of genes in the input space is about 17,000, whereas in GeneFormer it's 4096. The masking strategy for the models on the left only use non-zero genes to mask, but whereas in SC Foundation we use both zero and non-zero genes in the masking strategy. They also hugely differ in the training size, whereas CBIT is a very small model, only trained on 1 million single cells, whereas Informer and SC Foundation are trained on 50 million, 95 million, which are huge data sets. So essentially, for more details, I would refer you to the corresponding papers, but these are the pre-trained models we use to get the contextual gene embeddings from foundation models. Now, coming to the graph learning, once we define the graph as a set of nodes, which are genes, and of edges, which are regulatory interactions, we use graph neural networks to learn the node level representations of genes using multi-hop message passing. In these graph learning, each each node or each gene is updated using aggregated message passing through the neighborhood of the genes. So in this formula, M represents the message passing mechanism, which is different for different graph neural network, and ND represents set of neighboring nodes, and each of the layer graph GNN layer, the node representations are updated based on the multi-hop message passing. So we employ three different popular graph-based learning GNNs in this study. This one is graph convolution GCN, which uses convolutional networks to aggregate the neighborhood information. Sage uses sample neighborhood and then aggregates information from this sampled neighborhood. Uh, and Graph GAT uses attention on these neighbors to aggregate the information for each node. And after employing the GNNs, we get structure-aware gene embeddings, which are also the node level embeddings in the graph. So once we get these gene level embeddings from foundation models and uh, and the graph learning, we propose a unified gene representation that combines the representations from SEFM and GNN. The idea is that this unified representation contains both structural information and contextual information, which is very important in deciding deciphering the complex interactions between genes and cells. Um, so after getting the combined unified gene representations for each genes, we use a link prediction layer, which is essentially a fully connected layer based on MLPs to perform the link prediction. So for each pair of genes, it's either there is a link present or not present, and that's the kind of training data we use for doing the link prediction task. And we employ binary cross entropy loss to optimize our model. And as you can see, the back propagation using this joint graph learning is only happening at the GNN layer level and not at the foundation model level. So to validate our framework, we use B-line benchmarks, which constitute 7 small data set at various sizes from various cell types. And the ground truth we use is a cell type specific chip sequence. For the evaluation metrics, we use AUROC and AUPRC metrics. Um, as you can see in the results, our our framework with the informer showed average improvement of 7.4% in AURC and 18.6 in AUPRC, and all the values reported are averages from 50 independent evaluations per set. So in all the 7 cell types, SC-RegNet outperformed the baselines. And as you can, as expected, SC-BIRD being a small model uh, did not perform as well as the bigger models as SC Foundation and GeneFormer, which have trained on a very diverse and huge set of single cell transcriptomics. Um, I've also done application studies to decide which component to decide which component is contributing to the performance. And when we removed GNN encoders or SCFM encoder, the performance deteriorated, showing that the unified representation is what is helping the performance. Um, We also performed robustness studies because inherently we wanted to check if the bias in the training data is being either is it being propagated or not, or is it getting better. So to introduce noise in the training data, we sort of flipped the labels of ones and zeros in the graph. And we noticed that as we kept increasing the noise, SC-RegNet still performed better than other models, showing that even when the graph neural networks have some kind of bias, the contextual information is helping still giving us accurate results. We also did a case study to see if SERegnet is inferring biologically meaningful interactions, and by filtering out all the links which were predicted with greater than 90% probability, SERegnet identified 109 novel interactions in the human embryonic stem cell context. So, for example, we one of the interaction that was predicted aligns closely with the established rules of this factor in pluripotency maintenance, which is supported by the experimental literature. For even more further details, I would ask you to refer to our paper. To conclude, our comprehensive evaluations show that SERegnet consistently outperforms traditional and graph-only approaches, which underscores the potential of leveraging large-scale pre-trained models and contextual information along with the prior biological context. We acknowledge the main limitation of our model, which is the heavy reliance on the ground truth network, which may not always be available for new cell types, which is why our ongoing work focuses on or addresses this issue by incorporating the graph topology or knowledge of existing interactions, by injecting this knowledge into the foundation model parameters, by looking at whether the injection should be done at the input layer level, mid-level fusion, or the late fusion. Um, I just want to thank my advisor, Dr. Xuan Wang, and my collaborative advisor, Dr. Yu Wang, and Dr. Yisi Wang for sharing great, useful comments. Here is a QR code to our paper. I would also like to thank our supporters, and thank you so much for your attention for today.
Thanks, Sindhura. So for the sake of time, we will take one question. So is there one question online, or do we have anyone asking for a question? I see everybody moving around. Okay. Then in that case, I will be the one asking one question. So since you're using the graph, the GNA for getting the structural information, have you tried different basic network, like I'm sure you use PPI, but have you tried other biological network for that source of information?
Sorry, have I tried what? Could you repeat that?
The structural information in your model is coming from, I suppose, the protein-protein interaction network. So, but of course, the performances will depend on which prior biological network you're using. So have you tried different inputs?
No, that's a good question. Thank you. So currently, we've only used prior GRN networks, but in our future work, we are considering diverse networks like PPI networks, gene ontology networks, and other prior biological networks as well.
Okay, thanks, Ken.